Hi, so this is my last lecture in the series of 3 looking at conservation. So in lecture 1, we looked at some of the human-mediated threats that are causing extinctions around the world, about the protected areas that we're trying to use to prevent those extinctions and why they don't perform as effectively as they should. In the second lecture, we looked at why particular species are vulnerable to extinction and the knock-on impacts that has for protected areas, and again, why our unbiased understanding of particular species may lead to conservation programs that aren't as effective as they could be. And finally, in this last session, we're going to continue to focus on species-focused work, and we're going to look at how small populations, which is one of the main problems in conservation. So how do we estimate the minimum populations required for sustainability into the future? We're often in conservation working with small populations, which have been caused by a lot of the threats we talked about in the very first lecture. So if habitat degradation and loss has caused the species range to decline, we often have to conserve small numbers of individuals, so small populations. So we're going to look at what the problems that can cause, and how you can predict the extinction risk of those populations and whether or not we need to be worried about those particular populations going extinct in the near future. The learning outcomes of this are to look at the problems that are caused by small populations. Look at how you monitor a particular small population. So this ties a little bit into the exercise that went along with lecture 2. So we were thinking about how you can monitor the banteng and how you can assess their population sizes, and how you can design a research program to better understand that species. And then in this lecture, we're going to have a look at some other methods of how you can monitor small populations, especially when they're difficult to study, when they're shy or other things. We're then going to look at how this population monitoring can be used to estimate important dynamics of that population, so rates of breeding and rates of mortality. And then we're going to show how those estimates of breeding and mortality rates, which are here called vital rates, can be used to predict future population growth and the risk of extinction. So when we're looking at small populations, this is often called the small population paradigm. So these are the problems that are faced by small populations just because of the fact that they are small. So small populations face greater risks than larger ones, just due to the fact that of the small number of individuals that are in that population. There are 3 main areas of risk associated with being a small population. And these things are often called stochasticity. So as populations become smaller, they become more at risk of chance events that cause problems for those populations and can increase the risk of extinction. So the first one is genetics. So I think this will be covered in some of the other lectures that are being recorded. But when populations are small, it is just because there are fewer individuals, it is likely that you have lost some of the genetic diversity that was present when the population was larger. And because there's less genetic diversity in that population, genetic diversity is the kind of the raw material that evolution acts on to adapt and gives rise to adaptations to particular environmental conditions. So therefore, if there's less variability present, it means that in the future, if the environment continues to change, which it is very likely to do due to climate change and human impacts, there is less scope for that population to adapt to those changes, to be able to adapt. So if a lot of that genetic variation has been lost, then it can't be used for that population to select for particular advantageous diversity, advantageous adaptations going forward into the future. And there's also likely to be more inbreeding. So inbreeding is when related individuals mate with each other to produce offspring. This is a problem because with the natural diversity, genetic diversity present in populations, some individuals may well have deleterious mutations. So these are genetic mutations that have a negative impact on fitness. However, if they're present at low levels in the population, or if they're not kind of very, very deleterious, if they don't cause death immediately or something like that, then they may persist in the population at low levels because they don't cause negative enough impacts to be selected out of the population entirely. They can also be what are called a regressive adaptations. So they only exhibit their deleterious, their negative consequences if an individual has two copies, one from each of its parents. However, so it may be that some individuals in these populations have these negative mutations. If you are mating with a related individual, it is much more likely that that related individual has the same negative mutations that you have, if you have any at all, than an unrelated individual would have. Therefore, inbreeding can cause problems because it can give individuals two copies of those regressive mutations, but it can also increase the chance that the offspring have these negative mutations. So it can start to fix those negative, those deleterious mutations within future generations of the population. This picture here is this butterfly from the UK called the Glanville fritillary. And this is the first study that gave strong evidence that inbreeding in small population sizes can give rise to negative effects for the conservation of a particular species. So for this butterfly, mutations that were negative to its health started to be fixed in the population, and survival rates started to drop and breeding rates started to drop as well. So it had real world consequences for the conservation of this species because the population was small. As well as genetic issues, we also have demographic issues, demographic stochasticity. So this is stochasticity that's just based on chance events in the mating and the population dynamics of a particular population. A lot of this can be caused by, as I said, by these chance events. So say that a certain number of individuals don't breed every year just due to the fact that they couldn't get enough, just due to chance. So the fact that they didn't manage to store enough resources, kind of eat enough to store enough resources to be able to breed that year, or just the fact that they by chance didn't find a mate or something like that. So, you know, it's not just deterministic effects that cause variation in breeding rates, it can also just be chance things. In a large population, the fact that some individuals don't breed every year due to chance isn't much of a problem because it's likely that lots of individuals will. However, in a small population, if just by chance for one particular year, lots of individuals don't breed, that has a very important impact on the population because it's a much higher proportion of the total population that therefore isn't breeding. So that's just one of these chance things that can happen. The other is sex ratio bias. So whilst probably on average for most species, there's a 50% chance that an individual will be male or female when it's born. That definitely varies for different species. Some have their sex determined by temperature, such as sea turtles and other things. But if we just take that as kind of a general rule of thumb, however, that will obviously vary year to year, just depending on chance. So it may be that a certain year that more males are born than females. Again, in a large population, that probably doesn't have much of an impact on the population as a whole. However, if, say, there's only 10 individuals born in a particular year in a small population and 8 or 9 are male just by chance, then there are very few females coming into the population, which can cause problems in the future if if that happens a few years in a row, it may be that there aren't enough females to then produce young to sustain the population. So again, these chance events. Another thing is allele effects, which I'll actually talk about on the next slide. Yeah, I'll talk about on the next slide. But again, that can be problems that are caused just by the fact that a population is small. The final one to talk about here is environmental stochasticity. So this can be chance events in the environment that cause problems for populations. Again, all populations will be affected by environmental stochasticity. So say there's a drought a particular year in a savanna, so say in the ecosystems that I work in, say there's a drought, that causes low amounts of food available for herbivores, so there isn't much grass available for the grazers because it doesn't green up in the wet season because there isn't enough rain. Then that year there's poor food resources, which may cause weaker individuals to die, may increase mortality rates, and may lead to low breeding rates. But if there's a large population, especially if they're spread over a large area with different habitat types, it may be that only a small part of that range is affected by the drought, or that even if it kills a few individuals, it doesn't have too big an impact on the population as a whole. However, if that particular species has been reduced to a very small area, that drought may cover the whole range of that species. So it may affect all of the individuals. And also, if it's killing, again, say 10 individuals from that population in a population of 1,000, yes, we're losing some, but we're not losing a high proportion. But say there's only 20 individuals, because our population is really small, we've lost half of our population. So it's very important to the future of the conservation of that species. Small range sizes can also be affected by extreme weather events. So not just droughts, but say a whole species lives on a particular small oceanic island, is endemic to that island. That island is affected by a cyclone or a hurricane. It could very easily wipe out that whole species in one go. Whereas if a species is spread out over many islands, we can lose the population from that one, but we have these other ones to kind of fall back on. So this is some of the problems that we get with small populations and why focusing on them is so important for conservation. So this is just outlining some of the allele effects, which is again one of these things that can affect small populations just by the fact that they are small. So generally in population modeling, population studies of population dynamics, we have a population growth rate. We have a population size that increases up to a carrying capacity, which is here K. So K is the number of individuals of that species that a particular habitat can sustain due to food resources, water resources, a whole range of other things. Generally, if there's no allele effect here, the population growth rate starts at 0. If we have no individuals, the population can't be growing. But it's positive all the way. It increases up to a maximum, which is about, we normally think to be about half of our carrying capacity. At that stage, density dependence starts to take effect. So individuals within that species are competing for resources. So it starts to reduce the population growth rate. Because that competition can reduce the amount of resources that each individual gets, it starts to increase mortality rates in the population and decrease breeding rates. The population growth rate therefore starts to decrease but remains above 0 up to the carrying capacity. The carrying capacity, the average population growth rate is 0. So the number of individuals dying equals the number of individuals born. It won't stay exactly that number. It might go up a bit. But if it goes above the carrying capacity, the average population growth rate becomes negative because of that density dependence. So it pushes it back to the carrying capacity. And the same if the population decreases. So say there's a drought, it kills off a few individuals, but that means that we're below the carrying capacity, so the population could start to increase again up to K. However, allele effects can be are things that happen when a population is at a small population size. It starts to either decrease the population growth rate with a weak allele effect, or even make it negative with a strong allele effect. So say our population generally exists at quite a low density, which we'll talk about with the Sumatran rhino on the next slide. So say there's individuals only come together to mate. If we lose lots of individuals, and the individuals we do have are very spread out, very far apart, then if they exist at an even lower density, then they can really struggle to find mates. There aren't enough individuals around for them to easily find a mate when it comes time to breed. So with these strong early effects, such as the difficulty of finding mates in that example, when we get to a certain population size, the population growth rate actually becomes negative. So if we get to a population size that goes lower than the point where that red line crosses the x-axis on that top graph, the population becomes negative. The population growth rate becomes negative, and it pushes the population towards a size of 0. So it pushes us towards extinction. So this is another example of why small populations can become very vulnerable to extinction in certain circumstances. So the Sumatran rhino was thought to be affected by these alley effects. So back in 2019, the last Sumatran rhino in Malaysia died. It's now just restricted to Indonesia. But it was thought that this species was existing even in the absence of human impacts. It exists in quite dense forest. It's solitary, except for mothers and calves, like we see here. And they only came together in times of when the female was in estrus to breed. As these animals were hunted and as they lost habitat, their densities decreased further, and it became very difficult for them to find mates. So it's thought that the difficulty of finding mates was an allele effect experienced by this population. Population growth rate went below 0, and it starts to push it towards extinction, accelerating the decline and possible extinction of this species. Okay, so now we've talked about some of the problems associated with small populations. How do we actually estimate population growth and population, if it's so important to the risk of extinction, population size, and the population, the change in it over time, so the population growth rate? How do we actually estimate that? So we can just go out and count individuals, but are you actually managing to observe all of the population? It's probably unlikely. So you may be just getting a portion of that population. And are we able to estimate how much of the population we're actually measuring when we do that? When we do this, we also need to take into account whether or not we're sampling across the whole range of a species. So say, as in the first lecture, when we were talking about how a lot of our research is focused on protected areas, even though they cover a relatively small part of the globe. Say we just study a species in a protected area. It may be that the density there is higher than in the disturbed habitats outside the protected area. So if we count the individuals in the protected area and then extrapolate that estimate across the range, so we find a density of 10 individuals per square kilometre, and we multiply that by the whole known range, we may get a misleading estimate of the population size because the population density might be much lower outside of that protected area. Therefore, we're overestimating the population size. So another way of so if we can't observe the whole population in one go, another way of trying to estimate population size is to find individuals repeatedly, and to do this thing called mark recapture, which I will get onto an example of in a second. So these are just two examples of how we can sample population sizes and how it's context dependent. So say we're studying elephants in a savanna, we can use, because they're very big and they're in open habitat, we can use things like planes and binoculars to go out and just count the number of individuals because they're relatively easy to spot. Again, you have to consider the fact that it's unlikely that the whole range of this animal is in the same habitat. So do we need to factor in the differences in densities? And also, do we need to, you also need to make sure that you're not counting the same individuals over and over again. So if you go out on repeated days, you need to make sure that you're not just counting the same ones every day you go out. However, say we're trying to study this snail species here, which is very small. You can't just go out and count them all. It's much harder to find. We can do this mark recapture study design where we get an estimate of the population size. And to do this, we can use this equation here. So the capital N is the total population size, which we're going to estimate using this calculation. The capital M, is the number of members of the population that are captured initially and tagged. So we go out on day one and we capture a number of individuals and we tag them. Okay, that's how we get capital M. And then the lowercase letters are the estimates we get when we go out and we do the same. You have to do exactly the same experimental design because this method works based on a ratio that you're capturing on the assumption that you're capturing the same proportion of the population both times. So the lowercase n is the number of members of the individual that are captured when you go out your second time. And the lowercase m is the number of members of that on day two, the number of individuals that you capture in your second sample that were tagged. And then we can then use this equation to try and calculate the total population size. So I'll go through an example because I know it can just be confusing to look at maths like this. So here we go. So this is an example with tortoises. We go out on day one and we capture 10 individuals and we mark them with this red dot. When we got on day two, we captured 10 individuals again, exactly the same experimental design, has to be, because we have to make sure our sampling effort is standardized. Of that second sample, 5 of them were marked. And then we can then plug these numbers into our equation to calculate the total an estimate of the total population size. So where our capital M is 10, because it's the number of individuals caught on day one, our lowercase n is 10. It's unlikely that you would get exactly the same number of individuals on both days, but just for this example, that's what we've got. The lowercase m is therefore the number of tagged individuals, the number of individuals with red dots that we caught on day two. And then we calculate our total population size using this equation here, and we get a estimated total population size of 20. So this is an idea of how we can we can estimate the population size of a species that is difficult to measure the population size directly. So when we think about this, have a think about your Banteng study design, which was the activity from the second lecture, and think about perhaps how you could use this in your estimate of the Banteng population size. So with a large vertebrate, it's quite difficult to directly capture and mark them. But if you're using a camera trap study design, can you identify particular characteristics of each individual? You can count them. And then if you did 2 different camera trap surveys, and you could identify a certain number of individuals from the first study in the second one, you could then use this to try and estimate the population size as a whole if you're struggling to directly measure the whole population. Once we've estimated our population sizes, we can then use this to try and model our population growth and estimate what our extinction risk is into the future. So when we're doing this, one of the primary things we want to do is estimate the population growth of our target population. So this is R, which is the birth and immigration to our population minus the deaths and emigration we see. So this is expressed as a percentage. So if we have a population of 100 black rhinos, say, over a particular time period, we see 10 births and 5 deaths, but there's no immigration or emigration because they live on a fenced reserve, then R is 5, and then this is expressed as a percentage. So it's R is 5 in a population of 100, so we have a 5% growth of our population. This is often done annually, but it can be done over whatever time period you want, as long as you're clear about the time period you're working with. This is just a diagram that shows the basic units, the basic rates that determine population size over time and population growth rate. So we have birth and immigration that add to a population, and then we have deaths and emigration that take away from it. So the cumulative impact of all these together gives us our population growth rates and allows us to estimate both that and the population change over time. So when we're modeling once we have these vital rates, so if we can estimate what the birth and immigration rates and the death and immigration rates are over time. We can then use this to try and estimate how our population is going to change into the future, which is very useful for conservation, because if a population is going to decline, we want to be able to act on that before it happens. So if we can predict that's going to happen, we can try and tackle it before it becomes a problem. So when we're doing this, we have to construct a model of our population. This is often done by splitting a population into different age brackets. And this is based on, we kind of group individuals of similar ages that show similar birth rates and death rates. So this is an example from a black rhino population model. So the first year of a vertebrate's life is often very important because they often have very high mortality rates because they are predated, they can be vulnerable to diseases and other things. So the first year is often occurs by itself. And then in this black rhino population, we have two pre-reproductive life stages. We have that 0 to 1, and then we have 1 to 5. 1 to 5 is separated from the other ones because after 5 years old, rhinos can breed. So for 0 to 1 and 1 to 5, the fecundity rates will be 0. So those are pre-reproductive. So those become individual, they become groups because they have similar birth rates of 0 and death rates. And then above 5, we have these three reproductive life stages where death rates and reproduction rates are similar. So 5 to 9, 9 to 17, and 17 to 33. And then we have one post-reproductive life stage, which is older than 33. So individuals survive past that, but they don't often breed. So we split our population up here into different groups based on similarities and vital rates. Once we've done that, we can then construct a Leslie matrix, which is a set of numbers that express the birth and death rates for each life stage. So this big life stage in the middle, this big matrix in the middle, sorry, contains each of these vital rates. So ignore the small matrices on the left and right for a minute, just look at this middle one. Across the top, we have these F numbers, and these are the fecundity rates. So these are the per capita average number of female offspring reaching N0 born from mother of the age class X. So I know that's a bit confusing, but basically, so each column in this matrix is one of those age classes. So if we're looking at this population model here, we have 6 age classes. And so F0 would be the number of 0 to 1-year-olds that are giving birth, which is obviously going to be 0 because it's a pre-reproductive life stage. But that would be the birth rate for that age class. F2 would be the birth rate for this 5 to 9 age class, and they are able to breed. So that F2 would have a particular number, and that's the proportion of female individuals in that age class that would be breeding every year. Okay, so that's how the fecundity rates go into our Leslie matrix. And then the S numbers, which go along the diagonal of this matrix, are the proportion of individuals that survive every year. That's both survive that survive from time t, which is the year we're in now, to time t plus 1. So this Leslie matrix assumes that each age class is only 1 year. So if an individual survives, it has to progress to the next age class. If we look at our rhino model, that's not exactly what we're doing here. So the arrows at the bottom, where it says S2 brackets 1 minus G2 or similar, those are the number of individuals that are surviving but staying in the same age class. And because they're in an age class for more than 1 year, individuals that survive will either stay in that age class or will transfer to the next one. But in this Leslie matrix, it assumes that every time an individual survives a year, it progresses to the next age class. So once we've constructed our Leslie matrix, which includes our survival and fecundity probabilities, we can use that to project our population into the future. And what we do is we use this Leslie matrix in combination with a matrix that shows our population structure for time t for a particular year. So on the right-hand side here, we have these numbers n0, n1, down to nw minus 1, where w is the maximum age that an individual can reach. So the numbers in here are the number of individuals that are in each age class. So say for our rhino population example, n0 is the number of individuals in the 0 to 1 age class at time t, so in any particular year. Then to project how our population would change into the next year based on our survival and breeding probabilities, we multiply the Leslie matrix with this matrix on the right, and it gives us the one on the left that has the number of individuals in each age class at time t plus 1, which is just the next year. And that's represented here. So we multiply A by NT, which is our population at time t, by our Leslie matrix, and we get population at time t plus 1. Now to do this, we have to do a multiplication, but matrix algebra, so the way you multiply matrices together, is a bit different to how you would do it just with numbers. So to get our time t plus 1, our population size at time t plus 1, which is our matrix on the right here, we multiply our Leslie matrix, the big one on the left, with the one in the middle, which is our population size at time t. So to get the number at the top here, 39.3, you multiply 0 by 45, then you add that to 1 times 18, you add that to 1.5 times 11, you add that to 1.2 times 4, and that gives us our, once you added those four numbers together, it gives us our number 39.3 here. So this just gives a summary of how we do this multiplication to get that final number. So you add, so you times every number on the top row by every number in that second matrix there. So this way, so top left is multiplied by top, and then you move across one on your big matrix and down one on your small matrix. Do the same again, do the same again. So you do that for every row of your Leslie matrix. You plus all those numbers together, and it gives you your population at time t plus 1. So we've recorded that we're 45 of our youngest age class in our population, but we predict that in time t plus 1, so the next year, we're going to have fewer of those individuals in that age bracket. So this is our estimation of what our population structure is going to be in time t plus 1. So in this population, there's only 4 age classes. One is pre-reproductive, so you can see that the top left number in this matrix is 0. So no individuals in the youngest age class are breeding. But in the second 3, fecundity rates, so in the fecundity rates for the other age classes, all those 3 age classes all have a probability of breeding. Now, the numbers here can be above 1 because this species can have more than 1 young per year. So say with a black rhino, it can black rhinos only produce 1 young a year, so this number couldn't go above 1 because it can't females can't have more than 1 calf at a time. So this number for black rhinos would be below 0 because only a fraction of individuals would be producing 1 calf each. However, this species can produce more than 1 young at a time. So on average, each female is producing 1 or more than that young per year. And then this species can only live, if we're assuming that each age class here is 1 year, this species can only survive to 4 years old. So for the first age class, 80% of individuals survive to age class 2, which is the 0.8 number there. For age class 2, only 0.5% survive to age class 3. For age class 3, only 25% progress to age class 4. And when they get to the end of their age class 4, all individuals die. There's no probability that individuals can survive past the age of 4. So that's the kind of structure of this population. And that's how we then use our vital rates and our current population estimate to predict how our population will change into the next year. And then you can just do this over and over again, and you can model how the population is going to change into the future. This is then really useful for conservation, as I said, what uses can there be for this, because we can predict how different populations are going to grow or decline into the future. So we can compare different populations, we can identify ones that are at high risk of extinction. So we can, based on current conditions, current vital rates. The other thing we can do is that we can try and predict the effect that conservation interventions would have on a particular population. So say we identify through this modeling that a population is at risk of extinction. When we do that, we can try and say, well, what would happen if we try and improve survival of a particular age class? So say we see that a lot of juveniles are dying. We say, well, if we can intervene and prevent juvenile mortality, say it's often caused by an invasive species. So we predict that controlling this invasive species will reduce juvenile mortality by this much. Will that have an impact on the population? Before we actually conduct the intervention, which can be expensive, and if we find out that it doesn't work, then we've potentially wasted that money. Instead, we model what effect that would have on the population. And then if we can see that it probably won't have an impact on the conservation of the species, we can try and model other interventions until we find one that we think would be useful for the conservation of this species. So it saves us time and money by predicting what the effectiveness of conservation interventions would be. So the methodology of using these population models to predict extinction risk is called population viability analysis. So we're looking at how viable different populations are. It requires a lot of data, which can be difficult to come by, especially for wild species. And it can be overly pessimistic. It can often predict declines when in real life that population might be sustainable. However, it does provide evidence for management and can be surprisingly robust. And it can be used, as I was saying, to evaluate a range of different management scenarios. So this is an example of a population viability analysis that was done on elephants in Myanmar. And you can see how we can model what impact different conservation interventions would have on the population. So our baseline population modeling is just looking at vital rates as they currently are in the population. So the measured death and birth rates. And as we can see, over 200 years, we predict that our population will be about the same level as it is now in the future. So it might be stable. A lot of population models, you see how at the start of this, we kind of get ups and downs. A lot of them have these things called transient effects that are kind of just mathematical artifacts of the way the model was set up. So it predicts kind of short-term variations in growth rate, which may not occur in real life. But if we just want to look at the long-term effects, the long-term growth rates, they're not too important to look at here. Just if we look at the long-term growth rates and the long-term impacts. So for our baseline, our current vital rates, we predict that the population won't have grown over 200 years. However, if we want to think about whether or not our conservation should focus on birth rates or mortality rates, we can use this model to compare that. So we look at the green line. This is modeling if there was a 10% decrease in juvenile mortality rates, which we were talking about before, can be important for which can be very high for particular vertebrate species. So that gives us an increase over 200 years, but that increase isn't as big as if we were going to focus on adult birth rates in this population of this species. So this population model would suggest that for conservation to be the most effective for this species, we would want to try and focus on increasing adult birth rates. Now, how we do that is an entirely different question, but it does show, without actually having to carry out the conservation intervention, which may be one, expensive, and two, take a long time, we can predict what is the best thing to focus on for our conservation efforts. So, quick summary: small populations are vulnerable for genetic reasons in the long term, and also but can also be vulnerable for other things in the short term, so demographic stochasticity and environmental stochasticity, as we said. The IUCN, which is the International Union for the Conservation of Nature, but also researchers and other people, can use population models to establish extinction risk criteria. So the population modeling methodology that we run through very quickly here is used by international conservation organizations to assess extinction risk. So the IUCN, which produces the red list, which is kind of the go-to resource for assessing extinction risk in particular species, they rely very heavily on these methodologies and similar ones to try and assess extinction risk. And they come up with their critically endangered, threatened categories. Yeah, and then we can just calculate basic population growth based on total numbers as we did, or we can try and use more sophisticated population models to try and predict how populations will change into the future. So the activity for this lecture isn't a go home and do it yourself one, because these population models are quite complicated to set up and instigate, and they require specialist software. A lot of software is free, but you just need to know how to use it to be able to run these models. So along with this lecture recording, I'm going to upload a recording of me running through one of these practicals on a software called R, which is free to download, but requires a bit of experience and know-how to be able to use. And I'll just show how you can use these Leslie matrices, very simple ones, to predict how populations will change in the future, and how we can use them to predict the impacts of different conservation interventions that focus on different vital rates into the future. So thank you very much. That's my last lecture. I hope these 3 lectures have been useful. They've given you a nice introduction to some of the basics of conservation, how human impacts are leading to extinctions, and how we can tackle that on a kind of a landscape scale and also on a species level scale. Thanks very much, and I hope they've been useful.